Hey guys, welcome back to another new video by Biology with Zangshan. So today we are going to do the 0610/42 May-June 2023 Paper 4 Theory Extended. So this is the Paper 4 that I've done for my June 2023 series. So today we are going to discuss everything that has come to this paper. Okay, let's go to question 1. Figure 1.1 is a diagram of part of a cross section of a leaf. Okay, so guys, remember that the paper is in the description below. Please check it out, yeah? Okay, for part A, explain why a leaf is considered to be an organ. Okay, so they're asking you why is this an organ. So because there's like a group, because it is made out of groups of tissues which are working together to perform a specific function. So this is how you get your 2 marks. This is basically the definition of what is an organ. Definition of organ. Okay. Part B. Explain how layer B and layer C in figure 1.1 are adapted to their function. So they want to you to explain about their adaptation. So you can see layer B and layer C. Okay, however, when I was doing my doing this paper during the exam, I had a misinterpretation for layer C. Why? Because it could either be the spongy mesophyll cell, or the vascular bundle. Soon out, I found out that it's acceptable that vascular bundle and spongy mesophyll cell can both be mentioned. Okay, so how you're going to write this answer is by firstly stating what is layer B and what is layer C. So B is the palisade mesophyll cell and C is the spongy mesophyll cell and the vascular bundle. So in layer B, the cells are tightly packed, so light energy can reach to the cell for photosynthesis to happen. It also has many chloroplasts in each cell. In layer C, the cells are loosely packed to allow gas exchange to happen and diffuse. The xylem helps to transport water and mineral ions, and the phloem transports sucrose and amino acids. So that's how you get your 6 marks. So this is 6 marks, just basically stating the layers and then explaining the adaptations of it. Okay. Question 1C. A student placed a plant in a very hot room for 12 hours. There was a bright light in the room and the plant was not given any water during the 12-hour period. Figure 1.2 shows a series of sketches that the student made of the stomata during the investigation. Okay, so you can see that this is basically located in the stomata. So part 1, identify the cell labeled D on figure 1.2. These are the guard cells. So the guard cells are part of the stomata. Part 2, state the main function of the stomata. It allows gas exchange. Okay. Part 3, state the advantage of the plant of the change to the stomata shown in figure 1.2. So what is the advantage of the stomata changing this way? So it helps to reduce water loss from transpiration, because if the stomata doesn't change, all water will lose out easily, which can cause wilting eventually. Okay. Part D. The student increased the humidity in the room and repeated the investigation. Predict and explain the effect of high humidity on the stomata. So when there's high humidity, there will be lower transpiration rate. So the whole point is just what they want you to expand from here. So the stomata will take a longer time to close, making reduced water vapor diffusing out. This helps prevent the guard cells from losing water so that it becomes and remains turgid. Okay, that's 3 marks. Okay, question 2. Figure 2.1 is a diagram of part of a DNA molecule. So it shows figure 2.1 here. So part A, draw a circle on figure 2.1 to identify one pair of bases. So this is asking you for one pair of bases. It could be any of this. It could be this, the one that I circle here. It could be here, here. Anything that is joined, like between A and T and C and G. Okay? Okay, part B. The percentage of T bases in the DNA of a species is 29%. Calculate the percentage of bases that will be base G in the DNA of the species. Space for working. Okay, how do you solve this one? It's actually very simple, because we need to know that A and T, since they say T is 29%, but this is only for one. Remember that T also binds with A, so A and T, they are together. So they need to have the same amount of bases. So it's to be 29 plus 29, which is 58. So since percentage is out of 100%, we use the 100 minus 58, we get 42. So 42 is the total of C and G in total. So how can you find G? Is by 42 divide by 2, you get 21%. So the answer is 21%. Okay? So if you never show you're working, you get 0, even though your answer is correct. Okay? Okay, question 2C part 1. State the name given to the length of DNA that codes for a protein. Gene is the answer. Okay? Question C part 2. Explain how proteins are made by a cell. Okay, this question is basically asking you to state what is protein synthesis and the process of it. Okay, so the mRNA is first copied from the DNA, it stays in the nucleus, and mRNA moves to the cytoplasm. The mRNA passes through the ribosomes, then it assembles the amino acids. Lastly, the order of the amino acids is determined by the base sequence of the DNA. So where the marks come from is from 1 mark here, 2 mark here, and the 3 marks here, 4 marks here, and the 5 marks here. Okay. Okay, question C part 3. DNA controls cell function by controlling the production of proteins. State 2 types of cell membrane proteins. So this question I also struggled on, so I also got it wrong. Now I know what a cell membrane protein means. It means that it's basically proteins that actually work as like a key function to something. So remember synapses? The synapses, it looks like this. Okay, then it has like a bunch of receptors like this. So the receptors here are acting like a membrane, a cell membrane protein, where eventually it will bind together onto the protein receptors by the neurotransmitter molecule. So this is what it means by cell membrane proteins. Okay. Question 3 part A. Antibiotic resistance is an increasing problem worldwide. Erythromycin is an antibiotic. Figure 3.1 shows the daily dose of the erythromycin per 1,000 people over a 13-year period. So you must see that they already linked it. It must be in per 1,000 people. So the number of bacterial infections resistant to erythromycin per 1,000 people is also shown. Okay. So part 1, calculate the percentage change in the number of bacterial infection resistant to erythromycin per 1,000 people between 1993 and 1995. So from this, they want you to find percentage change. Remember, percentage change has to be either positive or negative depending on the result. So since they want to see between 1993 and 1995, you need to compare. So between 180 to 100. So percentage change, this shows that from 1993 to 1995, it has already decreased. So this has to be a negative percentage change. So the formula for this is initial minus final over initial times 100%. So 180 minus 100 will be 80 over 180 times 100 equals to 44.4444, and using two significant figures, which is just minus 44%. So the drop from here is a total of minus 44%. Okay. Question part 2. Describe the data shown in figure 3.1. So they want to describe the data in this. So the daily dose of erythromycin is peak at 1989 at 2.8 doses per 1,000 people. So there's a 2.8, which is the peak. Always identify what is the peak here first. Okay. The bacterial infections is peak in 1993 at 180 bacterial infections per 1,000 people. Then there is no record of resistant infections until 1991. So after this, there will be a lower record of resistant bacteria. Okay, so this is how basically you describe the data by identifying the peaks, the lower points, and the trends of the graph. Okay. Part 3. Suggest reasons for the change in the number of bacterial infections resistant to erythromycin from 1993 to 1995 shown in figure 3.1. So if you see that there's a change in number of bacterial infections, it must be some sort of awareness already. So there is a use of a fewer doses of erythromycin and people are more aware of the overuses of antibiotics. Okay. Part 4. Explain how bacteria become resistant to antibiotics. So this question is actually very vague. So the point of this answer is to link to natural selection. So there's a lot of key parts to natural selection here. So how you explain is that this process is by natural selection, where random mutation occurs in from some bacteria, giving rise to genetic variation to allow it to better survive and reproduce. Then those survive bacteria passes on their resistant allele to other bacteria, making it more adaptable to the environment. Okay, this is 5 marks. Okay, question 3, B part 1. Bacteria are prokaryotes. State 2 features of all prokaryotes. So cell wall and cell membrane. There are a lot more. Circular DNA. There's also a lot more. So this is not just the only answer. You just have to state 2. Okay, question B part 2. Some bacteria have a flagellum. State the function of a flagellum is for movement and swimming. Okay. Alright, question 4a. A student recorded the information about an aquatic habitat. Figure 4.1 shows the student's notes. So aquatic plants provide food for freshwater shrimps and midges. Mayflies also feed on aquatic plants. The mayfly provide food for salmon. Stoneflies feed on midges and are eaten by salmon. Salmon also eat freshwater shrimps. Herons also feed on salmon. Question for part 1. Construct a food web to show the feeding relationship described in figure 4.1. Do not draw the organism. So what it's asking you is to draw a food web out. So just follow the instruction. This is how it's supposed to look like. Okay, so you can pause the video and also look through back the question again. Okay, question for part 2. Complete table 4.1 using the information in figure 4.1 by identifying the names of the missing trophic levels and one organism at each different trophic level. So in the producer will be the aquatic plant. So basically you just refer back to your diagram, which is much easier to see. And the primary consumer will be the mayflies, the freshwater shrimps, or the midges. And the secondary consumer can be the salmon, or the stoneflies. And then the tertiary consumer would be the herons or the quaternary. Okay, for quaternary, it means like the fourth one. You must remember the word quaternary. Okay, not fourth consumer, quaternary. Remember this. So herons and also can be considered as a quaternary consumer because first one, second one, third one, and the fourth one. Okay. Okay, question part 3. Outline how energy in the primary consumers in this aquatic food web is used to produce biomass in the secondary consumer. So, okay, whenever I also struggled in this question, thinking about like what does it actually mean. But honestly, after doing this, I realized that produce biomass, I just need to think of how does it produce energy and how is it going to be transferred, to different organisms in this. So primary consumers contain chemical energy because they are the first one, and which is transferred to the secondary consumer. So the glucose available, which is from their food and everything, is for respiration or used for growth, and maybe the synthesis of protein and the storage of glycogen. So it's basically kind of like just saying that how energy is useful in from primary consumers to the secondary consumer. Okay. Question part 4. Human also eat salmon. Predict the impact on the feeding relationship shown in the over-harvesting of salmon. Okay, you see, what I do is that I'll use a pencil during the exam, and they say what happens if this decreases. If this decreases, the herons will decrease. This will increase the stoneflies, the mayflies, the freshwater shrimps, and there will be lesser midges, but however the aquatic plant will still remain the same. Okay, so how I write my answers is that there are fewer herons as they have less food to feed on, and this causes the increase of mayflies but no changes in the aquatic plants. Okay, like the basically how many aquatic plants are there, it still will remain the same, it doesn't change at all. Okay, question B, describe what is meant by the term decomposer. So decomposer means by the organism that gets its energy from dead organic material. Okay, part C. Animals such as salmon can be farmed for meat. Explain why is it more energy efficient for human to eat crop plants than to eat livestock and have been fed on crop plants. So as there's fewer trophic levels, right, making plants becoming more energy efficient because it has the most energy. So going up to the food where energy becomes more inefficient. So as the trophic level increases, they are lost by respiration, excretion through urine, and heat being lost to the surrounding. This decreases the energy as it goes up. Okay. Question 5. Figure 5.1 is a photomicrograph of a structure found in animal and plant cell. So first thing in the exam, I did my very big mistake is not seeing this animal and plant cell, because this to me just looks like a cell, but however it's actually a mitochondria. So state the name and the function of the cell structure labeled A. So this is basically a mitochondria. So the function of this is to undergo aerobic respiration or to release energy. Okay. Part B. State the two pieces of information needed to calculate the actual length of a cell structure A in figure 5.1. So what they're asking you to do is to, you see from this triangle formula, you need to know how can you able to find A, which is I divided by M. So I is image, or it could be the length of the cell structure, and M is just magnification. That's how you get your 1 mark. Okay, part 5C. The actual length of a cell structure A is 0.00075 millimeter. Convert this value to micrometer. So we know that 1 mm is equals to 1000 micrometer. So 0.00075 millimeter equal times by 1000. So this will become 0.75 micrometer. Okay, that's how you get your 1 mark. Alright, question 6a. A student investigated plant growth responses in roots and shoots. They used this method. Damp cotton wool was placed in 2 petri dishes. 3 bean seedlings were attached to the cotton wool in each petri dish. Each seedling was orientated so that the roots pointed in each different direction in each petri dish. Petri dish 1 was kept on its side in a fixed position. Petri dish was kept on its side and rotated constantly. Both petri dishes were kept in the dark and is conditioned for 2 days. After 2 days, the seedlings were observed. So you can see that in petri dish 1, they're in the damp cotton wool but is in a fixed position. However, petri dish 2 is rotating. So after 2 days, it will look like this, where it grows in one direction, and this is not changing at all. Okay. So in part 1, describe the pattern of growth of bean roots and the shoots in petri dish 1 shown in all 6.2. So in this one, you can see that they are basically growing. So all roots shows positive gravitropism and the shoots are growing upwards. So there are things that's going downwards, and some are going downwards, and some are going up. So both sides are positive. Okay, state the name of the growth response observed in bean roots and shoots, gravitropism. Okay. Part 3. Explain how auxin caused a difference in the pattern of growth shoots by the shoots in seedlings B and E shown in figure 6.2. So this is the 5 marks, right? You must explain that how auxin causes the differences. So you need to talk about how auxin is made first of all. So auxin is made in the shoot tip and diffuses down. You can say it undergoes diffusion, and this helps to stimulate cell elongation. Okay, this is the key word. So in seedlings B, there is a high concentration of auxin. This stimulates more cell elongation on the lower side of the shoot. So you can see that there is a higher concentration, so therefore it's going downwards. And in seedlings E, auxin is equally distributed, which stimulates cell elongation equally, allowing it to grow at a straight line. So you can see that because it is rotating, right? Rotating and then rotating there, auxin is equally distributed. So when you turn around, the auxin will still diffuse equally, you know. So it looks something like this, that is that there's equal auxins on the left and right. Therefore, it moves up and doesn't move there. It doesn't move to either side. It only moves up. Okay. So part B, seeds require oxygen and water to germinate. Part 1 state one other environmental condition that affects germination, temperature. Okay. Part 2 suggests why oxygen and water is required for germination. So that's my answer for it. This oxygen is for aerobic respiration and water is acting as a solvent. Okay. Last question, question 7. Complete the sentences about enzymes by writing a suitable word and phrases in each of the spaces provided. So this is fill in the blanks. Suddenly in this 2023 series, right, they always love to come out this, but it's actually quite easy, you know, in my opinion. So let's do this. So enzymes are involved in chemical digestion, which produces small soluble molecules that can be absorbed into the blood. Two examples of protease enzyme are pepsin and trypsin. Pepsin is produced by the stomach and requires acidic condition. These conditions are created by the release of hydrochloric acid, which provides the optimal pH for pepsin activity and also kills harmful microorganisms, or you can also say pathogens. Okay, both are the same meaning. The pancreas produces trypsin, or the small intestine if you want. Intestine produces trypsin, which breaks down protein in alkaline pH conditions. These conditions are created by a substance called bile, which neutralizes the gastric juices and also has an important role in the emulsification of fats and oils. So each correct word will give you 1 mark each, and you get a total of 8 in this question. Okay, so this is the end of the paper. So if I look back, I was quite shocked that there's nothing human in this paper. So it's all very plant-based questions, everything. So tricky. This is very unexpected to me. So after this, I think I feel kind of most importantly is to review my mistakes always. So hope to see you guys in the next video. Bye bye.